Hello everyone. Welcome to our channel Biological Sciences. Biological Sciences is a platform where we study and learn about different field and in detail about different concepts of biology and nature. Here we also prepare for the exams which have biology and different fields of biology as the core. So here I bring you the video related to an experiment and this video is about to find the source of contamination that occur in a flask of nutrient broth exposed left to the air. So let's see. So here we have to find out the source of contamination, right? So first of all, we have to understand what is contamination and that occur in a flask of nutrient broth. So we have to also understand what is nutrient broth, which was left exposed to the air. So what is nutrient broth? Nutrient broth is the general purpose medium that is used to grow desired microorganisms. So medium, that means a source or medium in which the microbe can grow, but we are talking about the desired microbe, the microbe which is beneficial for us, which gives us the product that is beneficial to us. So this medium will act as a food for the microorganism. It contains all the nutrients like nitrogen, carbon and phosphorus. All the essential nutrients will be present in this medium, in this nutrient broth. So if you have worked in an microbiology lab, then you must know that before culturing the microbe, we must prepare the medium which contains all the nutrients, essential nutrients for the growth of the microbe, and we have to prepare it in the sterile condition. We have to prepare in the sterile condition, otherwise contamination may occur. Contamination means the undesired microbes. Sterile condition means there will be no other microbes present until we provide the desired microbe to grow in this medium. So we have to maintain this sterile condition and prevent this contamination, the undesired microbes. But what happens is sometimes contamination may occur in this medium, and we have to find out the source of contamination, that from where this contamination could come. Okay. Next is we have hypothesized. What we have hypothesized is that pre-existing microorganisms present in the air contaminate nutrient broth, and this hypothesis is called the germ hypothesis. So uh, from this we can say that in air some microorganisms already present that could contaminate the nutrient broth, and from this we could predict that sterilized broth will remain sterile if the microorganisms are prevented from entering air. Like you can see here, this is sterile broth. We have sterilized this nutrient broth and there is no microorganism present here until it is sterile. So if we prevent, or we can just say we covered this broth, the flask of this broth, then uh, there will be no entry of air, and this could prevent the microbes to flourish inside this flask. But if we remove this prevention with this, then air could go inside and microorganisms could come inside and grow in this nutrient medium. So we have to test this, that what will happen in both the condition, right? So this is the hypothesis and how we have predicted that. Now the other hypothesis is known as spontaneous generation hypothesis. Here living organisms, we hypothesize that living organisms will spontaneously generate from the non-living organic molecules in broth. So I told you that sterile nutrient broth, the nutrient broth contains organic molecules like nitrogen. It contains nitrogen, it contains carbon, it contains phosphorus, it contains many other nutrients, and those organic molecules will help in the generation of microorganisms. So from this we could predict that organisms will spontaneously generate from organic molecules in broth after sterilization. So what will happen if this hypothesis is correct? Then if we cover this sterile broth, then also the chance is that microorganisms could generate in this broth spontaneously. So we have to test this. And so to test this, we have used what? We have used, sorry for this. We have used what? We have used these swan-necked flasks to prevent the entry of microorganisms, or we can say the air. We prevented air to enter inside this swan-necked flask, and to ensure the broth can still support life, we can break swan neck after sterilization. So how we will test this? Just look at this. So we have this flask. We have sterilized by boiling the broth. So heat can destroy the undesired microbes which will already present in the nutrient broth or in this flask, and after sterilizing there will be no microbes present here, and this condition will remain sterile. Okay, so we have two conditions, that we have unbroken flask. So we have unbroken flask. So what happens is there will be no entry of air, so it remains sterilized. Next is we have broke. So if we break the swan neck of the flask after sterilization, what will happen? It leads to that it leads to the entry of germs or microbes inside this flask, and uh, this broth, this medium becomes contaminated. So it gets contaminated because of the entry of air, and here we prevented the air to come inside this entry of air inside this medium, inside this flask. So the organic molecules does not lead to generate to generate the microorganisms, but the entry of air lead to the entry of microorganisms. So here is the result, that no growth occurs in sterile swan neck flasks. When the neck is broken and the broth is exposed to air, growth occurs. Conclusion is, growth in broth is of pre-existing microorganisms present in the air. So we have understood that uh, air, the pre-existing microorganisms present in the air, leads to the contamination of the broth. So thank you everyone for watching this video. I hope you liked this video. I hope you understood the whole experiment. If you have understood and liked this video, kindly subscribe, like and share so that other people also get the benefit.